Hi, we're back in a lab with a new sample now. We've got a piece of the lower crust from within a shear zone in Fiordland, New Zealand. And this old piece of crust here used to be a diorite and then has undergone many episodes of metamorphism and some deformation. Now we've taken a thin section of this sample from the shear zone and analyzed it on the SEM by a new technique, EBSD, electron backscatter diffraction. So EBSD allows us to use the beam to measure the orientation of a crystal at a single point in space. And if we move the beam ever so slightly on a big grid, we can end up mapping an area of the thin section. And it's extremely powerful because it allows us to determine what minerals are present, what shapes their grains have, where their grain boundaries are, and if there's any deflection or deformation, distortion of the lattice within those grain boundaries. So it's a tool for mineralogy, petrology, and structural geology. So I'm showing you some data from this sample here. This is what's called a band contrast image. And that means, in EBSD lingo, the quality of the EBSD pattern. The brighter the grayscale image, the brighter the EBSD pattern is, and you have a really big contrast between the bands that are diffractions off the mineral lattice versus the background. So you can see that this does a pretty good job of showing you where the grain boundaries are. In between grains there is no lattice, and so the contrast goes down to 0 because it's just dark. So check out right here, right between these two grains, there's a bunch of little black pixels. No contrast, that's a grain boundary. So this grain gave slightly worse patterns than this one here that's white. So this is one way to display the EBSD data. Now I'm going to show a different sort of map, same data set. Instead of band contrast, we're going to look at phase color. So every distinctly different color on here is showing you a different mineral phase. So we know exactly where the distribution of hornblende is in blue. Plagioclase, this is a composition of plagioclase in the green here. We've got some other, let's see, index minerals, here we go. Apatite in this location, lots of different things in this rock. There's also quartz in here too. You can see quartz in the dark blue. So that's how we look at the distribution of phases, and because we're getting multiple pixels per grain, we can also do things like measure modal percentages. Now another part of EBSD that's really powerful is crystallographic orientation. That beam measures the orientation of the lattice with respect to like its A, B, and C axes. And so that's pretty powerful because we can make maps of that orientation too. I'm going to show you a map of crystal orientations. So this is a total party confetti map here. It doesn't show you which minerals are which, it just shows you grains that are distinctly different in orientations than others. Here you can see things like twins. These are twins in the plagioclase grain. There's a couple more grains with twinning in them, very common in the feldspar. But you can see that even though there's maybe about 5 phases in this map, there's lots of different grains in here. The grain size is actually quite small. So it's a very powerful way of collecting data. Now this is all raw data, and what we have to do is take the raw data and process it into something that's more usable. So we're going to switch over to the processing PC in the other corner of the lab, and I'm going to show you this same map but processed, and then we're going to manipulate some of that data. So here's the exact same map, but this is the processed version. In purple, you're looking at hornblende. In green, you're looking at plagioclase. The red lines within the plagioclase are twin boundaries. The orange is apatite in here, and the maroon color is the quartz. There's a couple other minerals in here, but we are going to really focus on just the plagioclase and the hornblende in here. Grain boundaries are shown in black on this map. So this gives us a good idea of which phases are present, the sizes of those grains, and also grain shapes. So notice here that there is, in the purple grains particularly, an elongation to them. They're longer in the horizontal direction like this than they are in the vertical direction on the map. When we have mineral grains that align to have a certain shape, they've got an aspect ratio, we call that a grain shape preferred orientation. And just looking at this, we can see that it has one. But it's also useful to quantify what that shape preferred orientation is. We measure it like on a circle, 360 degrees. What is the dominant orientation of the grain shapes? Now, because EBSD is both a petrology, mineralogy, and also a structural geology tool, we can look at the grain shapes and then compare it with the actual crystal orientations. We have that data embedded within the data set from the SEM. So I'm going to show you some diagrams. They're called pole figures. And you'll have to remember a little bit of mineralogy to navigate the pole figures. I'm going to show you the orientations of the A, B, and C axes of the hornblende in these lower hemisphere projections. It's like looking into a cereal bowl, and if you had a pencil, where within that bowl the pencil dot would hit. That's the orientation of the axis. These are what are called pole figures. Here's the lower hemisphere bowl. You're looking down into that bowl, and these are dots that represent the orientations of the A-axes from all of these purple grains, the hornblende grains. This is the A-axis plot. These are two B-axis plots in here. This is an upper and a lower plot because hornblende has pretty low symmetry. It's monoclinic. Here's the C-axis plot over here. So we can see that the A, B, and C axes are plotted here. And now we're going to look at one grain in particular. See this red flashing light in here? That's highlighting the orientation of one grain. This one here. You can see a little red cursor right where my arrow is. And if you look over here to the right, this is a schematic drawing of how that grain is oriented in terms of A, B, and C axes. So red, green, and blue axes correspond to A, B, and C. So the A axis is roughly vertical, B is kind of coming back out at you, out of the page at you, and C is pointed this way, fairly close to this grain shape preferred orientation. Now this grain that we're visualizing here, we can see exactly how this plots up in pole figure space. Flashing little cursor here showing us the A axis is almost vertical. You can see that in the crystal orientation projection. The B axis is almost coming out of us here at the page. And then the C axis, flashing over here, is elongate in the horizontal way. So if we plot one point for every single hornblende grain in this map, that's what we're looking at here in the pole figures. 271 data points plotted here. Now you can see that those points, they're not randomly distributed. They tend to cluster a little bit. And that's called a lattice preferred orientation because we're looking at the lattice, the A, B, and C axes. So that's crystallographically controlled. Now we can measure how clustered these things are by contouring this data. So I'm going to use the software to do a little contouring of this data, and boom, here's what my pole figures end up looking like. We can see that the patterns are not random. Everywhere you see red, that's like a little set of bullseyes showing you that the density of the points there is the highest. So the density of A axes are highest right here with the red parts. The density of B axes is sort of around over here, close to the center of the pole figure. And the highest density of C axes are on these 2 points here, pretty close to horizontal. Now we can measure how close to the horizontal and vertical directions these maxima, or the bullseyes, are located. So in the case of the A axis, we're going to use this handy little measurement tool here and measure from the center of that cluster to the vertical direction. There we go, and that ends up being an angle of about 24 degrees. So the A axes have a dominant direction about 24 degrees counterclockwise from the vertical direction on the map. So vertical on the pole figure here corresponds to this direction on the map. Let's do that for the C axes. Now this is a little bit more tricky because the clustering is a little more diffuse, but if we pick the center of this cluster and we measure from that to the horizontal direction, we get an angle of about 20 degrees over here. So the C axes are about 20 degrees counterclockwise from the horizontal direction on the map. So this is your quantitative crystallographic preferred orientation. That's where the bulk of your A axes and your C axes sit with respect to the horizontal and vertical directions of the map. Now you can compare that with your calculated grain shape or fruit orientation. For the hornblende, they're going to do with a separate software program. So you can compare shape preferred orientation with crystallographic preferred orientation and answer the question, is the elongation of the grains linked to a crystallographic direction of the hornblende or not? And if it is, that tells us something about the process that occurs to give that foliation to the rock. Okay, have fun calculating.